Good morning. I'm Victor Martin, and I'm going to present the poster Minocycline-loaded chitosan nanoparticles, an intracellular approach for periodontal disease treatment. To begin with, periodontal disease consists in the presence of bacteria within periodontal tissues, provoking a strong inflammatory reaction called tissue destruction and ultimately tooth loss. Currently, more than 11% of global population is affected by severe periodontal disease. In this context, we devised chitosan nanoparticles loaded with minocycline, which possess antimicrobial and anti-inflammatory activity, to serve as drug carrier and to be used in situ. In addition, being at nanoscale, this material should be able to be internalized by the cells, tackling intracellular bacterial reservoirs. Nanoparticles were produced by ionic gelation technique using chitosan solution and TPP. Human gingival fibroblasts were used to analyze nanoparticles' intracellular trafficking, cytocompatibility, and inflammatory modulation. Regarding the uptake and intracellular trafficking, nanoparticles enter in the cells via micropinocytosis and endocytosis. After 12 hours, nanoparticles were found inside endosomes and lysosomes, and the number of vesicles was increased in comparison to control. Then, after 24 hours of incubation, we can see autophagosomes and amphisomes, the fusion of endosomes, suggesting an autophagy induction. The cellular trafficking is the same as P. gingivalis, one of the main periodontal pathogens. PG can survive inside the lysosomes and then escape to cytosol or even to neighboring cells. When these nanoparticles follow the same pathway, they can deliver minocycline inside the lysosome, and this functional autophagy activation can prevent PG from escaping. Moving on to cell viability, nanoparticles did not provoke cell death, and no differences regarding cell morphology were detected, despite the autophagic induction. Finally, to analyze inflammatory modulation, we stimulated cells with LPS for 24 hours prior nanoparticles incubation. Cells were also incubated with minocycline solution. Control is represented by the line at one value. As we can see, nanoparticles downregulated the expression of these four inflammatory genes more efficiently than the minocycline solution. To conclude, nanoparticles are capable of inflammatory modulation without hampering cell viability and follow the same intracellular pathway of pathogens, making them a potential parasite. Thank you for watching, and I invite you all to check our paper with these results. Cheers!